I actually was
Cool.
going to write up a bunch of questions for you, but then I went over your Google Talk twice,
Yeah.
and I was
Yeah.
like, this man has laid out enough, to fill it all in. But I wanted to let everybody know, please go check out his Google Talk. It's absolutely wonderful. Anybody listening to this in the future, go Dr. Christopher Mason Google Talk, lays it all out very beautifully. It's excellent. We are going to talk some about that because I do want to talk about your book, obviously. Let me tell the
Yeah.
audience a little bit about what you've been doing. How's that sound?
Great. Sounds great. Sounds good.
All right.
Yep.
All right. So in 2013, guys, Mason launched the PathoMap project to create the first genetic map of a city. That's pretty awesome. This study led to the establishment of the International Metasub Consortium. He also was a principal investigator for the NASA Twin study, very important study. He can enlighten us on that if he feels so inclined. He has also led the first demonstration of sequencing in zero gravity and designed the genomics and bioinformatics methods and experimental protocols that were used on the ISS, the International Space Station. So for the mission to sequence DNA in space for the first time. It looks promising for the Mars mission. I know you talked a lot about that.
Mm-hmm.
This work has also led to Dr. Mason's selection as the chair of the steering committee for the NASA Gene Lab Data and Sample Archives. It looks like, and has highlighted, I can't pronounce some of these, my friend, whale cornel medicine on 2 NASA patches. Okay.
Yep. See.
Yeah, man. Okay, well let's just get into talking. Can
Great. Great.
you give me, because genomics are probably what the most important thing about genetics, or what could you say to people listening in the future about that?
Yeah, so genomics is the collection of all genes. So people think of genetics like they have a gene. We often think of like a gene that gives you a risk for, say, diabetes, or makes genes make you tall, and because you can see tall parents have tall kids. But there's not actually just one gene though for height, or a gene for, say, red hair. There's usually multiple genes that contribute to make any part of your body, part of your cells. And so genomics is all of the genes together. So not just one gene or two, but really trying to understand biology and genetics by looking at every gene. So, genomics also can refer to technologies and methods to do sequencing of DNA, characterizing cells. Really, anytime you're doing work with DNA or even RNA, or even variations of genetics, it could be genomics, and in many forms.
Right. So Francis Collins, he's a genomicist, or what is that? A geneticist,
Yeah, you can say
and he
geneticist.
helped map. Yeah. He
The
helped
first
map the
genome,
human
yeah,
genome. Yeah,
yeah,
yeah.
yeah.
Um,
That's
can you
right.
explain to the audience chromosomes and how that differs from DNA, or how that plays a part in it? Because that is how I'm still a little confused as well.
Happily. So in DNA, most people think about DNA, that's their genetic code. It's in almost all your cells. And, if you think about it, you started as just one cell, as an embryo. Every one of us did. And all the instructions are there in that very first cell, all the DNA that you need to make every tissue in your body, your brain, your heart, your muscle, your lungs. So the instructions are there in that bit of DNA, but it's not totally naked. It's not sitting out. It's 3 billion letters. And so to actually package it in a way that can be copied and moved and condensed, otherwise it'd be too big if it was all laid open. It's packaged up into what's called chromatin, and then that becomes like bricks that you put together to make chromosomes, which basically is just a really big condensed version of all these 3 billion letters of genetic code, 3.1 billion letters total, into one small form that then you can keep in the center of a cell.
So DNA double helixes, they make up chromosomes. Would that be correct to say?
Yeah, there's a lot of them. Yeah, billions of them that make up chromosomes. Yeah, or
I'm
millions.
just
Yeah,
trying to make it work in my brain, man.
yeah,
Just
yeah.
yeah.
Definitely.
Uh, I liked how you talked. Let's talk. Let's talk about your book in the 500 years, because I love talking about that and how you went over all the statistics of. Oh man, I can't even remember half of it. It was so interesting. But like I said, I didn't want to just pelt you with questions. So, do you want to tell
Sure.
us what you're doing right now?
Happy. Yeah, and I put someone was kind of checking about the MC1R gene. That's actually one of the most well-defined genes that gives you red hair. That probably accounts for 80% of people who have red hair. They have a particular version of that gene. There's a few other genes that are probably at play, but that one's the most significant gene. That if you have that, you'll see a lot that more in actually like Swedish, Scottish, Irish populations. And some of that's because if you live farther away from the equator, you don't get that much sunlight. So any bit of sun that you do get, you want to have paler skin so you can get enough vitamin D. And
Yeah.
but anyway, so my book, just to comment on the chat there, but my book is called The Next 500 Years. I have another book coming out later this year. It's called The Age of Prediction. But my most recent book,
Ooh.
is basically a plan and a moral argument as to why we have to go out into the stars, and then a technical and genetic and genomics plan on how to get there. And so the moral argument is that we as humans are the only species that can understand extinction, so we're the only ones that are aware of the frailty of life. So it's really only up to us if we want to preserve ourselves, which is nice, but also all life as we know it, because eventually the sun will engulf the planet. The Earth will be basically completely eaten by the sun in
Yeah.
about 5 billion.
Yeah.
So if I just
Can't
put
have
those two
all our
things
eggs in
together,
one basket, can we?
that's right. That's exactly. We're all in one basket right now. And so if you put those two facts together, it's a simple conclusion. Like, well, if we're the only ones that are aware that life is in danger and life will eventually be completely obliterated by the sun, the only way that life will survive is if we act on it. So it's a really simple connection of those two facts, which I argue then gives me a duty to enact it. So how do we understand genetics and biology to potentially even modify it so that it could survive on other planets is basically the argument of the book. And then the book lays out in the next 500 years all of the things we can and should do to prepare for missions
Yeah.
to Mars or potentially beyond.
Well, we can talk more about that, or we can talk more about the ethics. I'm kind of interested
Yeah,
also in the
you
ethics
both.
of
Yeah,
this
yeah.
because, yeah, I know you are. Well, where to ask first? Should have written down some good ethical questions. I mean, what about CRISPR?
Mhm.
What about the ethics that come with that, or any genetic engineering? Right?
It's a big question because history is full of examples where we thought we did something. We were going to try and fix an ecosystem, like putting rabbits into different environments, and the rabbits take over, or invasive species. So CRISPR is a precise method for something that we've been doing imperfectly for a long time. So I think we know we can screw things up, right? So CRISPR, you could say, well, I'm going to edit the piece of DNA. CRISPR, for those of you who don't know, it's
Yeah.
clustered, regularly interspersed short palindromic repeats. Basically, it's a long term that just means it's kind of a bit of a scissors that can go and cut DNA and swap out pieces of DNA. And so it's basically a way you can edit genetics and play with them. And the danger, though, is what if you don't do what you think you're doing? You modify one gene, but it changes something else as well. Or what if it doesn't do what you thought it would do? So you could always do something wrong. But but this is actually true for all of medicine, right? We think we know what's causing your infection, so we try one antibiotic, but we might be wrong. And if it doesn't work out, you try something else. The reason people are more concerned about CRISPR is because you're changing your DNA, so it's harder to just fix it. What's important, though, there's different kinds of CRISPR that have been deployed in the past 20 years, and some of them are just like cut DNA and they break it. Others are what are called prime editing, where they only change. DNA is double stranded, so two strands like a ladder, and one of them you can actually just take out one part of it, change it, and then put it back. What's called prime editing, and it's much more gentle, if you will. There's also variations of CRISPR where instead of modifying DNA, you just change whether a gene is turned on or turned off, kind of like flipping on a light switch. But
Oh, okay. So what would that part give us technically in the let's extrapolate on this and have a theoretical conversation. What could we gain from that type of CRISPR?
what's cool is then you can, if you want to turn genes on just for like the weekend, like let's say you're going to the mountains and you want to turn on genes for hemoglobin or carry more oxygen in your blood, you could just do it for the weekend. So instead of taking a drug to change some of your blood chemistry, you could change your genes basically. And so that's
Wow.
what you can do is the chemistry for it is often it's called methylation, which is just a small group. So someone just posted that in the chat. Is a way you can epigenetically control whether something's turned on or turned off, and that is because you're
Okay.
changing the chromosome and the way it's structured. So those are all things that are called epigenetic, meaning on top of genetics. So you don't
I was
change the
about to
DNA.
ask, could you explain epigenetics for us, please,
Sure.
the best you can, because I'm a dummy when it comes to this stuff, man.
Yeah, actually, the best example is again, you kind of think about just the fact that we all started as the DNA that's in that when you're an embryo, we all started as just one cell, and then one cell becomes two, then four, then eight starts to copy to make. Today you have trillions of cells. Usually, estimate is that most adults have 100 trillion cells, lots of cells, human cells, microbial cells, a lot of cells. But it started as one cell. But if you look at your body, just look at your hands, your skin in your hand is different from your muscles, which is different from your, well, the cells in your mouth and what's in your brain, the neurons in your brain and your cardiomyocytes in your heart. So every cell type in your body is a different kind of cell, but the DNA is the same. So actually, the instruction set is there to make every kind of cell, but epigenetics is how you turn something on or off in different kinds of cells to give you this plasticity. So you can have one DNA code, but many manifestations of that code, many types of cells in your body, which is the epigenetics. You control when and where DNA is used, even though the code is the same. It's the same 3.1 billion letters effectively in all
Mm-hmm.
your cells. It's the same code.
Mm-hmm.
It's just you're changing what you turn on and turn off is kind of the epigenetic control.
Okay. Okay. So, well, that's really interesting. So let's what about some of the other CRISPR techniques you talked about? I forget now, you said in the center of the folding one, what was that? The where you lap it?
Where you let like
You
a
said
I think chromatin I was saying, or you said lap it. Say it again.
the first one you mentioned, and then the second one. The third one was turning the gene off and on, and then what was the second one you mentioned? I'm sorry. Should have paid attention.
There's methylation, and then there's also structure. Like you can change whether something is chemically modified or whether it's actually just open or closed. You do
Okay.
both.
Okay. What of those other two types besides just turning a gene off and on, what could we do? What could we
We
potentially
could
do?
Yeah, you could even you can add genes. You can actually, if like for example, if you can modify genes, add genes. If you want to get rid of a disease gene, like let's say you have something that's giving you sickle cell disease, you could actually get rid of the bad gene and replace it with a good one. Or you can even, one of my favorite recent examples is, you know, for people that have sickle cell disease, their red blood cells look like little crescent moons, and then the blood cells clump up. It can cause a big blood clot. So it's a horrible disease. But one thing you could do is, you can do a blood transfusion, or you think about it, you could replace the gene to try and get a good gene. But something that's also been done recently is instead of replacing the bad gene with a good gene, what they've done is reactivated a gene that is only present when you're a fetus. It's called fetal hemoglobin. So what you could do is just turn back on a gene that you used to use when you were a fetus and turn it back on as an adult. So then you'll see what's called fetal hemoglobin, but in an adult. And that's because it now has like a different version of the hemoglobin gene that you've reactivated, and that is a curative treatment.
Okay. That is super interesting. Oh my God, dude, I'm learning so much. I was just making sure we didn't have any callers, but I'm enjoying this conversation too much.
Oh, we have some
Um,
people on the chat are posting a lot. Someone talked about
yeah,
knockout mice.
yeah, if you want to go ahead and dive into the chat, go for it, my friend. Who
Sure,
we
yeah,
got here?
knockout
Let's see.
mice are when you knock out a gene, when you take away a gene. But knockout does sound like boxing, but it's not. Yeah, it's not that they're fighters. It's that they've lost something. The mice themselves have been knocked out. They lost a gene. Is what that means. Yeah.
This guy's unequilibrium says Linus Pauling revolutionized sickle cell research. I did not know
Yeah,
that.
he did. Also, yeah, did a lot of work in biology and in medicine. So he was really kind of a pioneer for sure.
Yeah.
Excuse me.
So your main training is in genomics and biophysics? Is
And
that
it's
okay?
basically how.
Yeah, well, I was just going to ask you because basically we're just explaining all this stuff for the layman out here, and I love this kind of stuff. My rooms are usually the group study rooms, so I kind of get to request people, and I wanted you to come on here because I liked what I saw from you on Google Talk. You seem like a really nice person, but
Yeah, thank you.
yeah,
Try to be. Try to be. Yes, yeah, try.
you're welcome. You're welcome. I appreciate it. So bioinformatics. What is important about bioinformatics and making that kind of software?
Yeah, so a lot of people think of software. They look at like your computer, you look at your laptop or your phone. Of course, that all runs on software, and that's for playing apps like games or Wordle or various games on your phone. That's all software. But software also has to be developed to analyze these genetic sequences we get in this genetic code. It's billions of letters, and then like I was saying, you have trillions of cells that generates these huge data sets. That the only way you can understand them and even process them is to have lots of computer science code and informatics code, which just means the means by which you can process and analyze the data is informatics. There's even chemoinformatics, there's geoinformatics for like chemistry or geology. There's different versions of informatics by different discipline, but bioinformatics just means these are the tools and the software we're building to look at molecules, look at genes, and understand what happens even in the hospital for medical records. So informatics is broadly any kind of tool set that's helping you process information. And we have a lot of it in biology because the data sets are stupidly large. So you need a really good code to understand them. Yeah.
So it's kind of like the methodology of how to make the software, of how to do the measuring and like, the metrics of it and things like that. Does that make maybe?
Yep.
Yeah.
And then and also deploying it and then getting it out on big servers like on cloud computing and big systems.
Right, to where it can. What about quantum computing? The quantum computer that IBM has, aren't they trying to do some multi-threading thing to where it allows us to watch how those genetic processes are going on? Or those, do you know what I'm talking about? Okay, I figured
Yeah,
you
yeah.
would.
So some of them, because at some point you're going to run out of computing power, and so then where you're going to start to get more of it is to go really down to the quantum level. There's even a group called quantum biologists. People try to think about, quantum fluctuations, where a small change of an electron spin could manifest and give you a pain in your heart or something. Really, it's kind of a more exploratory field, but it is this idea
Yeah.
that sometimes when it feels like you have like a spark of genius that happens, like where did that come from? Some of the ideas are like, what if it really is like a quantum fluctuation at the subatomic level that's creating these sparks of genius or a heart attack, kind of, which is still a bit exploratory, but it's an interesting idea of like, what if I could spontaneously, transport to another place, another
Well,
ideas. So,
David Bohm, I don't know if you ever heard of this guy, but David Bohm had those same ideas, about, well, it's not just one electron that would be causing, it'd be a series of like entanglement and superposition going on and all this kind of stuff to make, that thing you were talking about. But
yep.
yeah, so if we get quantum computing, smaller than a car, and we don't have to lower it to almost absolute zero to get it to work, do you see a future in being able to use that to
The lowering
process?
temps, that's one of the biggest challenges. Yeah, like getting liquid nitrogen, which is at like minus 300 degrees, and
Hmm.
yeah, so that is one of the challenges. But it's like a lot like fusion has just reached what's called the ignition point, where you start to get a little bit more energy out than when you put in.
Yeah,
And
not
I
the whole
think
system though, sadly.
no, it's not. Yeah, so it's still a ways to go.
Yeah,
Yes, you've been following it too, so it's
yeah.
exciting, right? But it's going to take some work and development and optimization to make it so it's really
Yeah.
ready for prime time.
We want that magnetic containment fusion. We don't want
Yep,
the
that's it.
inertial containment fusion. That's right.
Yes, yep, the
So
tomahawk
if
one.
the
Yeah,
whole if the whole system is using way more energy to put the laser in, you're like, okay, well, we got more out of that small piece of the system, but the whole system is still, is still taking up all this energy, and we don't, we're just not there yet, not yet. But I
yep.
have hope for the future. I'm like a futurist in a way. I love future studies. I'm
I'm an
not full in
optimist.
on
Yeah,
transhumanism, but I still think it's awesome to think about
yeah. I
transhumanism and CRISPR and all that stuff. Yeah, go ahead.
modifications. I actually think, even though I just wrote a book about the next 500 years, I don't consider myself a futurist, nor necessarily a transhumanist. I actually think. I just think I'm an empiricist. I think these are rational projections of what's likely to occur. A future, I just don't know what a futurist is. I mean, obviously someone who thinks about the future or wants to live in the future,
Yeah,
but I'd rather say I'm just an empiricist who has a decent guess as to what's likely, and then wants to. But the best thing to do is to actually create it. If you want, the best way to create the world you envision is to make it, right? So I think that's what I prefer to spend time on rather than just hoping or something.
exactly,
But
exactly. I feel the same way. Um, well, I was going to say something, but it left me. Anyway, so the quantum computing thing. Let's go on past that. Let's go to what what were the challenges? And I saw in the video what the challenges were to get this genetic sequencing on the ISS. What were
that
the
was one of
serious
the
challenges?
yeah, that was a hard mission because the device itself had to be flight approved, meaning if you want to fly anything in space, you have to do at least three things. You have to see what happens when it explodes, what happens when it gets caught on fire, and when you just smash it to pieces, are there components on the inside that are dangerous, like mercury or glass shards? So you have to really have what's called destructive testing approval. So that was the first thing. We just had to explode things and make sure they didn't explode in an extra dangerous way.
Yeah.
But to be fair, that was kind of the fun part. And we had to actually make sure that the chemistry worked. That we actually do, the molecular biology work where you have, enzymes, small molecules working, that they would work in zero gravity. And then we also had to get the protocol working and train the astronauts on how to actually run the experiment. Now, the advantage we had is that Kate Rubins was the one who went up for that mission, and she was a trained virologist, so she had already been in the lab. She had her own lab at one point, so it couldn't have been a better time to have her be the one that was going up to the space station. So so it took a
Nice.
lot of training, logistics, some hardware, and then also the informatics, the bioinformatics. We had to actually process the data on the space station, and also had to develop that for the first time. So it definitely was a lot of logistics and work to get it ready, but it worked. Now it's been up there ever since. So now it's a piece of the space station. So if you want to do some DNA sequencing up there, it's possible. It's part of the standard. It's kind of like making a phone call up there. It's just part of the standard package.
It's the same thing now as like going to the bathroom or whatever else they can sequence up there, huh?
Yep, it's just up there.
So let's talk about, since we're talking about empiricism, and I'm with you. I think you're using, Bayesian probabilities like we all are in some ways to see what's going to happen down the line. And I'm the same way. I'm a scientist at heart. You know what I mean? I want to follow that methodology, and whatever gets us to truth through that methodology, that's what I follow a correspondence theory of truth. I'm usually a scientific realist. Unless you catch me on a bad day, then I'm an anti-realist. But uh, no, I mean, let's talk about Mars. Let's talk about Mars for a second. What do we where can we go? You know what I mean? Like, where can we go now that we've done this on the ISS? Where can we go?
Yep. What's exciting, I think, is Mars isn't the best planet. It's obviously 1% atmosphere. It's very cold, a lot of dust. It's not hospitable, but it is the closest planet that we could get to and actually survive on. Venus is too hot. So it means that there's the lunar plans for experiments, Martian plans, but basically we're going to be setting up labs like these kind of sequencers that are going to be that we're in space. We're going to also put them on the moon and Mars, and that way, if someone gets sick, you can actually characterize their DNA right at that moment, and then actually see what virus is infecting them. Why are they getting sick? And so I think it's an exciting time because we have some of the hardware. To actually get us there, and also some of the informatics and the tools to sequence someone on Mars and say, oh, here's your problem, and then you can do an intervention right after that diagnostic.
What there wouldn't be any. Well, I mean, there would only be transmittable germs and stuff between people on Mars. There wouldn't be any viruses or anything on Mars at all, would there?
We hope not, yeah, because if there are, they might kill everyone. Right? So we got to see. Yeah, we'll find out.
We
Yeah.
find some buried
Probable.
civilization.
That's right.
Got
And then
wiped
last
out by
thing they
their own
wrote.
viruses.
Right.
Yeah.
Now, there is a group in NASA we work with them called Planetary Protection Group, which thinks about just this problem all the time of what's going to be moved between different planets. Someone said the movie Life. That was exactly the idea in that movie, is that there's a dangerous life form that ended up killing a bunch of people. So it was not good.
What was that one with was it Brad Pitt? Where there was this thing on Mars. They had gotten to Mars and it was just two of them. Anyway, it doesn't matter. I don't remember. If somebody remembers in the comments, hit that up, because I can't remember. It's either Brad Pitt, somebody really famous like that, and they had some kind of virus on Mars chasing them. Anyway, so can we go over a little bit more of your 500-year? I know it's a lot of stats, and for the audience, guys, please go watch his Google Talk, because it really covers all of this in a lot more strategic and academic and rigorous way than what I can cover it here. And I'm not the expert to be talking to my friend here. Somebody else could do a lot better of a job. But please go talk about this dude. So let's get into it. The 500-year thing. Let's go.
Yeah. So the first interesting. It started in 2010 when I first laid out the plan, and it was the idea that we just began to understand genomics. Basically, we barely knew. We had the first human genome, which is that 3 billion letter genetic code. We just had the very first one that we understood, but everyone's code is a little bit unique, right? So we're still finding out how many genes are in the genome, how does it work in different cells, how does it get regulated? So that was still being discovered. So I said at the time that we still have a good 10 years of pure discovery just about the human genome, because we don't even know what every base does. 3 billion letters, we know what maybe 2 or 3% of them actually do. So we had to figure out, what is all that DNA doing? Wow, where does it function? So I said that should take at least 10 more years, and we're now 13 years past that point, and we're still doing that work. So that's getting in now. We're in the second phase of the plan. That will continue. That basically will be a continued profiling of cells and genomes and using informatics tools to understand the body and what happens when you take a drug and what happens when you get sick. And then we're at the beginning stage, though, of synthetic biology, which is, for those who don't know, it's when you start to merge different pieces of organisms together, or even make entirely new kinds of life, because there's different kinds of life that doesn't exist at all. And because you can synthesize different kinds of biology. The same way you think of synthetic chemistry is when you merge molecules, or even just a synthesis of ideas. You put two things together that weren't there together. You synthesize them. So you can do the same thing with biology. You can modify it. You can have fruit flies taste like bananas. You can have oranges that glow when they get low on water. You can make all these kind of interesting changes to biology. And I say phase 2 is the very early stage, but then once we get to phase 3, we start to protect the DNA of humans and put them into space and see how well we can actually, instead of just doing physical protection against radiation, we could do it from the inside, like have every cell activate genes when it sees a danger potentially. And so that, I think at that point we'll start to do those trials and then start to put people into space. Phase 4, this 500 planet gets more into the next 30 or 40 years past now, where we start to actually have people get on Mars, start to actually live there and maybe even be born there after, say, 50, 60 years. And then it starts to get a bit more vague when you get to phase 5 and 6. That's when I start to say we're going to these ocean worlds, and we try and look at organisms on Earth that look like they could survive on, for example, some of these moons that are purely ocean in the middle of deep space, and then actually take lessons from them and then put them into human cells. So, for example, I mentioned, that we have all these genes that you could add genes or modify them. One gene that we don't have, for example, is the ability to make our own vitamin C, but we could put that back in. Or to make all of our own amino acids, we could add them in. Basically, make it so your cells can make more of the things they need. Because if you're on a long mission to Neptune, you don't have any places to stop along the way. So what if you could make every cell in your body have the ability to survive on its own and make all of its cells? It's kind of in the later phases. And then phase 8, 9, 10, just to group them. But basically, those later phases are when we start to get to the edge of the solar system. And then we actually start to I talk about having humans that could be part chlorophyll, part human. So they could be like plants, basically. Because if you have very little light,
Photosynthetic humans.
every photon you get could be energy. So I talk about that. So it does get a bit more exploratory towards the end of the book of like, but I do the math on, well, how much skin would you need and what would that look like? So I did the math on that in the book. It's kind of fun. But it gets more exploratory. But by the end of the book, by the end of phase 9, getting into phase 10, is when we say, okay, after 500 years, we should know enough to be able to put people on one spacecraft that could go towards another star. And I lay out all the different stars that we know about that we could send people to, that we could, in theory, survive today, because it might be liquid water, it might be warm enough, not too cold. But then, I say this is something we need, what's called a generation ship, where multiple generations live and die on the same spacecraft on their way to another star, which is a big ethical question too. Like, what if you're on let's say it takes 10 generations to get there, and you wake up and you're generation 5 and you're stuck on the spacecraft, and you think, well, I'm
Yeah.
stuck here, I have nowhere to go. Is it ethical for us to sort of imprison multiple generations on a small spacecraft on their way towards another star?
Yeah,
That
you
may
would
or
be focusing. I'm sorry. You would be
that may or may
focusing
not get
them.
you.
Yeah,
Yeah,
exactly. Go
they
ahead.
may not
Sorry
survive.
about that.
Yeah,
Yeah,
no,
they
that's
may not, or
exactly it.
they may hate it. They may be like, "Hey,
Yeah,
look, you have this Telos end goal for me to settle this M class planet or this Goldilocks zone planet, exoplanet, but I don't want to."
like,
you
what if
know?
they change their minds? We'd probably have to send multiple ships, because I argue that some of them might mutiny. They'd be like, well, to hell with this. I'm going to go somewhere else. Or they might, self-destruct, or, who knows. So,
Yeah.
but I take some solace in the fact that you think of the ships that would leave from Portugal or Spain in the 1400s. They they went on a very small craft, and a lot of them died, right? They went and they were never heard from again. And there is something that's very strong about the spirit of human, really just exploration. We want to go explore. We want to discover, and that's just native to humans. But then if you add on that, what I argued in the beginning is that this is actually a duty that we have to understand the universe, to serve as shepherds and guardians of life. That is something that we not only could do but should be doing. So that's the case I make in the book.
I agree. I agree that it's an ought and a should, that we should spread well-being to as many creatures and thinking agents as we can,
Mm-hmm.
and that all of that implies getting our eggs out of one basket and finding a way to survive in space and getting to Mars, getting to all these exoplanets. I like the statistics you brought up about the exoplanets. You know, we didn't know crap about them in what, 1990, and now we've
Yeah,
got,
first thousands of them. We got about almost 6,000 now
yeah,
exoplanets, which is a planet found around another star. Of those, about 400 of them are in the potentially habitable zone, meaning they might have liquid water, and they could potentially survive. Now, probably not guaranteed because you don't know what the atmosphere is going to look like, but it makes it so it's. I mean, but there used to be zero, right? There were zero exoplanets, and there were none known that would be potentially habitable. But now we have a short list of great candidates that are not that far away.
yeah. Do you think like silicate life or things like that would be invisible to us, or not? We wouldn't be able to tell that it's life.
We could look for it. So Sarah Seeger, someone at MIT, she does something called biosignature search, which is if you look at the atmosphere of different planets around the universe, can you guess as to whether or not life is there? For example, oxygen usually degrades quickly, so it's usually only there if something's making it, but not always. And so I think there is an ongoing question of like even ourselves, like what if someone looked at Earth's atmosphere from 200 million miles away? Would they know that there's life on Earth? Not necessarily, right? Depends on what they're looking at. So we may not even be able to recognize ourselves depending on how it's actually it's done.
No. I watched a video on this, how they see it, like they use the telescopes and stuff to see like what light shines through these planets, right? And then it's like the color of the light that comes through that they can see the makeup of the atmosphere. Is that similar to what you've read as well? Yeah.
Yeah, exactly.
Okay. Just
Because yeah, exactly. There's every atom that's there has this particular frequency and spectra that it emits when it gets excited. So it kind of defines what you'll
yeah,
see.
exactly. So I'm sure you got to talk a lot about that while you were dealing with all the NASA. Are you doing work with NASA right now? Are you focusing on your next book? What are you doing?
Yeah, both. Yeah, so I have a new book coming out in August. It's called The Age of Prediction, which is about. Here, I'll put it in the chat box here so people can see.
Please do.
Um,
I don't want you to give anything away because I'd like to read it, but I mean, if you want to share any of it, go ahead.
yeah, well, I actually think the one thing I might do. Let me just put this here. I'll copy this and put it in the chat box.
Please do.
Is I talk a lot about how AI is infused, almost every part of our lives and our society, and I think that's probably not surprising to anybody. That's here. Let's see. I don't know if that link quite worked here. Let me put it in the chat box here, but it's
Uh, let's see.
Let's do this. Hold on. I'm gonna put this in there. That might
It
work
went to
there.
the main page. Let's see.
So, talk about how there's predictive methods in medicine, um,
Yep.
and how, in the surgical room, you can predict what's the likelihood of someone crashing during surgery. You can predict which drug to give based on the sequence of a microbe. You can give someone the best cancer therapy based on predicting their DNA versus the drugs that are available. Um, and that's just in medicine. But if you look at what's present in self-driving cars, or look at airplanes that can fly themselves, or warfare with
Mm,
drones, autonomous drones fighting each other. AI and predictive methods are really defining really almost our whole lives. Uh,
yeah.
and so along the way, I talk about AI, and so there's different kinds of AI, right? There's the basic kind, which just does program yes, no, and
Hmm.
responds. There's
Strong and
basically five.
weak, all that stuff.
Strong and
Yeah.
weak, yeah, and that's what Elon Musk is scared about—the strong AI that's
Yeah.
self-aware, that could maybe kill us
AGI.
all, like the
Yeah.
Terminator movies, things like that, Matrix.
Yeah.
But
Yeah.
what I make the case for in the book is, well, what if there's a fifth kind of AI that comes after that, after the strong AI that might be self-aware? Because look at humans, right? We are self-aware. We look at you can have self-reflection, you can have theory of mind, you can think about what someone else is thinking, and we don't have a perfect track record, right? A lot of wars, famine, we've killed each other. It's not perfect, but
Yeah.
along the way, if you think that this idea of a species aware of its own extinction is a unusual feature, it may also be that this AI would appreciate that, would become aware of the fact that not just one entity goes extinct or dies, but that the whole species, all of the AI of a given AI species could go extinct. I think they will come to the same realization that we have, is that at some point, it's not just thinking about my own mortality, which is, of course, is a foregone conclusion. but the fact that you don't have to have a species mortality, that you can
Yeah.
persevere beyond the life of one sun. And also, if it does the same math we talked about in the beginning of like, well, I'm here today, but I might die. Well, I'll die at some point, and that the Earth might die. Well, then I have no choice but to think, how can I prevent? How can I stop that? If life has value, carbon based or otherwise,
Yeah.
and it's in jeopardy, then maybe we should preserve it. And I think they would
I
come to the
agree.
same. It would be the fifth kind of AI, like a self-aware, a
Hmm.
species-aware, really, of awareness of all life, not just of its own simple mortality.
Well, see,
I would hope.
I agree with you. I'm like an AI optimist. I think that if we raise them in spaces and treat them as we would our children, then hopefully they would be interested in the same things that we are and have the right to change their minds and things like that. And these AI, these strong AI that would be in the fifth level would have this kind of like super qualia, like this kind of like, I talked to Dr. Roman Yampolsky, and he told me, they would have this super qualia and they would realize that it was in the benefit of them to benefit us and all this. So, I'm, like I said, I'm very AI optimistic. I don't think Skynet immediately. What do you think when you think about that fifth level of AI?
Yeah, I think it will be. I don't think it'll be Skynet. I think it'll be like humans. There might be some, because it's also, to think that the AI would become a monolith is probably a fallacy, right? So humans are not all of one kind. So there'd probably be different flavors of AI, where some are maybe more aggressive, some are more peaceful. They might fight amongst themselves, right? So I think this presumption that we would be the largest perceived risk is probably not true. If there's any other sort of platform, we would probably just be, none of us are concerned about cockroaches taking over the world. They're a nuisance, but no one has views them as an existential threat. Same with almost any other species on the planet. We do, sometimes manage them, control them. Sometimes, of course, had an awful track record. Sometimes killed them all. But like the dodo, right?
Yeah, like the dodo.
But we now know that. I think what's interesting is, 100 years ago, there were no globally connected infrastructure and information systems. Like we didn't have the tools to map global problems. So we couldn't, of course,
Yeah.
solve a problem we couldn't even see. But now, I think we actually couldn't until the 60s and 70s. Like we solved the ozone layer in the South Pole. But until we could see it and communicate it at a global level, we couldn't address a global problem. So I give ourselves a little bit of credit, because when the dodos were killed out, there's no way to track how many dodos were left except by word of mouth, right? 100 years ago.
Hmm.
So I think that means we've just now got to the point where we can have these global goals, global challenges, and address them with a global effort.
I like
So I
that.
think it's good.
I think it is. I have more questions, of course, but we've got a friend of ours on here named Gator. He's a very smart man. He is a good skeptic, so hopefully he's got some good questions for you.
Love
Uh,
skeptics. It's
Gator,
good.
you are, yeah, I love skepticism, a healthy skepticism. Gator, you're on with us, brother. How are you?
Hi, gents. I'm
Hello.
well.
Oh,
Thanks
yeah.
very much for taking the call. Hi, Professor Mason. Pleasure to speak to you.
Pleasure.
I've got a couple of questions, but forgive me for a little bit of preamble, and also Thomas, if they're too long or the second one's too long, don't worry. The first one is about your insights and opinions on what I would simplify as saying regulation and control and governance and awareness of genomics. But if I was to try to explain specifically what I mean, it's about the way that technology filters through to the human race. So let's just take a smartphone. We're all
Mm-hmm.
users of smartphones. Most people just
Mm-hmm.
simply do not know what's going on inside the box, right? They use it,
Right.
and
Right.
that's the way it's meant to be for them. Now, the bummer is that, smartphones are actually remarkably powerful tools, much more powerful than the average Joe knows, and it's used in many nefarious ways that we have now come to understand. And
Sort of.
so,
Yeah. I
in
understand.
fact,
Yeah.
yeah, and one way one could look at these devices combined with the internet, combined with faceless people sitting behind that, is that actually we're now becoming controlled by our technology in many ways. One argument about why to ban TikTok kind of goes down this road. Um, who is it? Tristan Harris sort of makes the observation that we're at this technological inflection point in humanity where we are essentially children in a sandpit who are wielding the power of demigods, and yet we aren't really fully able to responsibly wield that power. Now, to bring this to genomics, I'm wondering what your thoughts are, because on the way that the average human and the majority of the human race, who have no real appreciation or understanding of genomic technology, and therefore are forced to trust the regulatory constructs, which are varied and poorly established in a lot of aspects of genetics, then end up ultimately becoming subservient users of the technology, where really only a very small number of people have been responsibly involved in development, control, regulation, deployment. And one of the, I don't think this is controversial to say. So, and the reason why I'll go to this level of detail is the example of SARS-CoV-2. It's now the GP160 AIDS insert, the furin cleavage site, and 12 sequence nucleotide, which is multi-trillion, one in trillions in likelihood of being naturally developed or mutated from the bat pangolin story, which has never been proven, basically puts us down the road of this is EcoHealth Alliance, DARPA related, diffuse, NIH funded genetic engineering, irrespective of how or why it may have entered the global circulation. All of these things get us into a question of regulation, control, and responsible development and deployment of genomic technology. How do what's your general feeling about this kind of stuff?
Yeah, let's get Mason, Dr. Mason here.
Yeah, sure. So, yeah, this is a big, I've done a lot of work on the diagnostics for COVID, done a lot of work here at the hospital. It's right across the street from me. When the first patients came through, it's been a saga, no doubt, to get through this. But there are, there are. I mean, so there's coronaviruses, which are sarbecoviruses. But there's also, there are other furin cleavage sites that can be found in kind of related species, like embicoviruses, which is a different kind of coronavirus that do have. So it's not like it never occurs in other viruses, and some of them are related. But it is this question of, how did it get there? I mean, so it definitely does exist in other kinds of viruses, multiple kinds actually. But what we do know is that from the description of the grants funded, their goal was to add the furin cleavage site, right, to coronavirus. So it wasn't, it was definitely part of the goals of what was some funded research. And so I think and it's actually a very solvable problem. If we went into the freezers in the Wuhan Institute of Virology and sequenced every virus and just said, here's the sequences, I could tell you in 10 minutes whether or not it was there or not. Now you could say, well, maybe they don't grab all the samples, they wouldn't do it. So some people will never be satisfied. But what I think is frustrating is, this is a very solvable problem, is to say, was it in those labs? You could sequence. I mean, we sequence viruses every day, and we can compare. I can tell you exactly who you got a virus from by sequencing the virus from one person, which would be genetically identical if you just got it from that person, or it may have only one small change. But we can tell how long a virus has been moving between people because it keeps mutating, right? So it's very easy to tell if it was there and then how long it's been there, but we just don't have the data. So and they didn't really collaborate or contribute to the data. But that's besides the point. I think I think I still haven't been convinced either way. I think because we don't have the data to make the full decision. It could have been engineered. I guess we know it's possible to be done. It could have been. There's other viruses that can and do have the furin cleavage sites. Again, there's hippocaviruses, novacaviruses. Those are the ones found. It's only found in bats, but it does occur there. So this cleavage site can and does occur elsewhere in other viruses. But I think the bigger point you're making, though, which I think is the important one, is that the ability to do these modifications is unprecedented. Like we could have an academic debate all day long about who did what, and without the data, we don't know. But we can say, well, we know what can happen, though, is that it could be done today. Like there was a grant funded to look at this and to add furin cleavage sites and to increase pathogenicity with a goal to understand it and, in theory, prevent it. But I think we know that it is an unprecedented world that this is not possible, that you can't quite do it all in your garage, but you can get certainly close. And I think it means that every bit of sequence information needs to be monitored the same way we monitor credit cards, right? Like if suddenly something weird shows up in your credit card you get a text message like in 5 seconds, right? It's because it's constantly being monitored. We know if something looks really unusual. The same thing is now happening with sequence information. For any genetic sequence, anytime you order something online, if it's too close to like an anthrax gene, it'll get flagged and say, would you tell us more about that order, please? So like it's getting to the point now where the companies doing the synthesis and creating the reagents are tracking it the same way we expect our credit card to be monitored for fraud. So I think that it's getting there, but it is a brave new world for sure.
Thanks. What about if we just sort of take a step down this road where there's a tension between the knowledge, involvement, capability, and competence of a regulator versus any practitioner or corporation acting with high levels of funding, high levels of power, and degrees of opacity. So, for example, that can mean that I think it's fair to say all regulators are always behind every curve, particularly when it comes to technology, because the nature of creating regulation is loaded in Western culture to position them into, or essentially a corporation is seeking to fully capture regulation. Frankly, is in so many industries have proven this is proven, and so an example of something that cure an 11-year-old girl. Now the funding for that was all public, but that treatment has been now taken up by, I think it's Moderna, but it may be another. I can't remember now. No, Novartis, and they are now charging $425,000 for access to that treatment when they didn't even fund the development of it. It's actually publicly funded, which is basically a gamification of the entire corporate regulatory model, particularly when you look at funding sources. How do you feel about that kind of construct and that possibility? Because that is really,
Yeah, it's so I think these are great questions, and there's Peter Kolchinsky is an old friend. If you look at this book he published called The Great American Drug Deal, it goes into some of these exact questions of like, well, yeah, if we publicly fund something, why is it so expensive later? Now, sometimes it's because things do cost money. Engineered cell therapies, for example, some of the systems that we have here at the hospital, the system itself costs millions of dollars just to get. Each of the therapies require many, many rounds of passage and modification, which is itself is $50,000-$60,000 just of supplies. So sometimes new things are expensive, but he makes the point in this book that there is a deal, if you will, between the public and the biopharma industrial complexes, that eventually things do go off patent and do come down in price. It takes longer than all of us would like, but he makes the case it's not as bad. I think the other thing that's at play, though, so there are costs. I'm saying that's one part. The second part, though, is there's a market and there is a for-profit system that is their number one goal is to maximize shareholder value. That is their fiduciary duty. They're not nonprofits, right? That's what we know, like the cost of medicines in other countries is much lower, and treatments. And so, once you get past the actual cost of something, which sometimes is high and it starts, there is a drive to make a lot of money, right? So and that's a big, it's a feature and a bug. It's like the feature of the system is we can rapidly iterate and get new therapies, but the bug is it's going to be expensive, and it might stay expensive longer than it should. Yeah, and then also you get some people that are just in it for the finance, and they're just trying to make more money. So, and they're all working together. So in the same boardrooms. So that's the pros and cons of capitalism in a way. But Peter in his book makes the argument that in the long run, he calls it the armamentarium of medicine, that we are getting more and more tools in the toolbox to treat current and even future diseases. And so he makes the argument that it's annoying and it's expensive, but in the long run that it is worth it because we're far better off today than we ever were for so much technology that we have. So it's an interesting book if you want to read, but he goes into some of the really egregious, excesses and abuses of the system, which there's many. So it's an interesting book.
Thanks. I've got one very quick other thing in case you don't mind, which is different. With people often think that our genetics DNA is static, but it's not, because obviously epigenetics is the kind of constant changing expression of gene expression, and it's more dynamic than perhaps people would understand. And also, we're now looking at DNA storage capability, so a gram for a petabyte or something like that. Yeah. Now, do you have any specific thoughts about whether we can combine DNA, a sort of memory storage? Because in a way, we can look at our own DNA and say there's a form of memory there, i.e., all my memory instruction set of how to be, how to function. But if I add another set of DNA, which I can freely read write to, and that's also containing now biogenetically available data, then essentially I might be able to carry the internet around with me in the form of
Yeah, or even there's these storage containers, ARC containers. I don't know if you've heard of these before, but there's very cool. They can actually store. There was the let's see.
All these links, by the way, everybody, will be saved in the chat for future. So please check all the links if you're listening to this video in the future. They're all very important.
I'm going to put one other thing. So it can be stored in DNA. There is something called this like storage crystal that was sent. Remember when the Roadster it got
like a genetic arc?
No, it's called the ARC Library, and it's a storage device that has a copy of basically everything in Wikipedia and most of human knowledge. When you threw it up there, and now it's orbiting around Mars, like this totality of human information in a small storage disk. But you can do the same thing with DNA. So Twist Biosciences, among others, are companies building DNA storage. And in principle, like if you look at if you just look on your DNA in every one of your cells, it has all the information you need to make everything in your body, and it's only in one tiny cell, right? So that's the idea behind it. And yeah, it's early days, but we're getting to that point where DNA storage might be better than a lot of the magnetic tapes fail after 5, 10, 20 years. DNA is good for millions of years, right? So why not use it? And so it is coming. It's a very exciting field of DNA storage, definitely.
Like genetic memory, right?
Yeah, exactly.
Making this like genetic, like he said, like this genetic construct that would allow access to electromagnetic field. Real quick, Gator, Dr. Mason, how long do we have you for, bud?
Oh, I have to run at 5. I can be a little bit late to the 5 o'clock meeting, but I can
Oh,
stay on
you're
for a few
fine.
minutes.
You're fine. I've pretty much asked all the questions that I wanted to ask, so I don't want Gator. If Gator wants to keep going, he can. Anybody else who wants to call in and talk to Dr. Mason, we won't bite. I promise. Come up and talk to us. I know there's not too many people in here. It's all of our regulars, but we're growing this app. We're growing this app, guys, and we want it to get bigger, and that's why we have people like Dr. Mason coming to talk to us, and we want people to get this.
Yeah, and I view it very much as a geneticist and a scientist. I feel like it's my job and my duty to get out there and tell people about the cool technologies that are coming out, and it's my pleasure to come visit. So, if any last question, happy to do one last question if you got one.
Gator, throw a last question in there.
Okay, if we look at how we're now at the stage of being able to correct the genetic or change something genetic, like for example, the fixing leukemia in this young woman after birth. If we were going to go down the road of basically modifying humans to say make their amino acids and vitamin C as you describe, we have 2 choices. Do we just one thing across all of your population if you only have partial knowledge of the future consequences? How do you kind of toss up of that?
So I think it'll be like we do like this is the first time in human history we've had even this question right in front of us, right? So I think we will have to do it like we do most medicines. We do it slowly in small groups of people, 10 people at a time, 100 people at a time. Like we didn't get a CRISPR cure for sickle cell and did it to every trials. We basically monitor people not just for one patient's life, but for their their progeny and multiple generations, to be extra sure that we haven't screwed anything up, because it's an extra powerful responsibility we have and technology. So the other alternative is you could have one continent where all the people stay unedited, like maybe Australia, and then we leave them alone as like a control group. It's another idea, but I think the reality.
Well, there are some people who believe that they are less. They have been less advanced.
That's right. So I think it'll be deployed slowly, like most medicines are, and carefully as it should be. But I think if something really appears, you have to say, well, this appears to be the pros outweigh the cons. This is something we could deploy more broadly. The biggest example, things like fluoride in drinking water, right? Like it generally helps with dentistry, or folic acid in bread. Like you can't even buy bread anymore that doesn't have folic acid enriched in it, because it seems to prevent neural tube defects. But there's probably a rare population of people where maybe it's a slightly higher risk, but that if for 99% of people, it decreases the chance of you having a deformed baby. The pros outweigh the cons, and then we just deployed it on everyone. There's no longer a choice to buy flour without it, unless you make it yourself. So it's interesting that there's precedence for this, what I call, that the acid to every loaf of bread they make. It would be too logistically hard, so we treat it at the source. But it does depend on the fact that you think, okay, this is we're so sure we're going to do it for everyone. But it's very rare in human history. But we're going to be at this one of those points where, like, well, we think this is really good. Maybe we do it for everybody. But I think that's decades and decades away, because it's such new technology, I guess. But but thanks for the question, and thanks for having me.
Yeah, thanks, Chris. I appreciate it. It's been a pleasure talking to you. Thanks very much, Thomas. Appreciate it.
You're welcome. Dr. Mason, thank you so much. Please come back anytime you want to.
Pleasure. Thanks for your interest in checking out the book, and a great group here. It's really wonderful. Thank you.
All right, guys. See you, brother. Thank you. All right, guys. That was fantastic. Awesome. Does anybody want to come up and talk about it, or should we go to Discord? Anybody go come up and talk to me? Hello? Nivik, I see you down there. Oh, we still got Chris in here. Hopefully. Well, maybe it's just still showing him. Hopefully it lets you leave the room, Chris, and it's not just bugged. Here we go. We got Gator. Gator!
Hey, Thomas, I really appreciate actually what Pangburn's doing on this platform, which is different to other, the people you've brought on recently are really just interesting and nicely varied, and they are also current. So I really think you're doing a good thing with trying to get more speakers like this, because just the ability to ask somebody of like Steve Keen's level of competence, or Professor Mason here, or Avi Loeb, questions directly is a really good thing. Thanks a lot.
Oh yeah, and you can tell Dr. Mason knows everything he's talking about, man. You can tell that is a professional through and through. He keeps up with things in physics. You know what I mean? Every good scientist should be as interdisciplinary as they can be. And when I find that in someone, I respect the hell out of it. You don't find enough multifarious autodidacts, But Gator, man, you have such good questions, and I really appreciate you always coming on and throwing some good skeptical questions in there. Are you on the Discord, Gator?
No, I've kind of registered an account, but I haven't really used it. I just don't really have the time to sort of go down that rabbit hole, really. So I just sort of dip in here from time to time.
That's okay. You're always welcome on my show. You know that. But if you ever feel like it, I think Travis can probably. Oh yeah, that's right. I can drop him. Hang on. Force mute, remove from speakers. That's right. Okay. I didn't want to keep it too much longer than this, guys, because I personally do not know enough about genetics. I studied a good bit. See, Travis just posted the Discord link for anybody who wants it. Please come and join us. But in the future, when we talk to Dr. Mason again, I'm going to have to do a little bit more studying into all of what he did. Only had like a day this time. And his Google talk was amazing. So if anybody wants the real rigorous, like numbers, statistics, graphs, go watch that, because that's better than I can do. I had a layman's understanding of this at best. So, yeah, guys, Gator, I guess we'll catch you. I may do a show later tonight, so if you want to show back up for that, you're always welcome.
Cool.
But
cheers, Thomas. Take it easy, mate.
Yeah, cheers, Gator. See you, buddy. Everybody, thanks for yes, sir. Everybody, thanks for coming to the Pangburn Hangout. Thanks, Dr. Mason, for coming and talking to us. That was fucking awesome. I get excited about this kind of stuff because I'm learning. Please come us in the Discord, and I guess if nobody else wants to come up and talk, we're gonna end this session. Thank you guys, and love you.